Welcome back to this next video, and in this video we are going to talk about the monoclonal antibodies. When you talk about the monoclonal antibodies, these monoclonal antibodies are abbreviated as MAB or the MOAB. These monoclonal antibodies are monospecific antibodies. So you need to understand this term monospecific antibodies. Now this term monospecific means specificity to a singular antigen. This mono means singular, and this specific is referring to the specificity. But this term monospecific is a very broad term, and it can be used in a variety of the meanings. For example, if the antibodies they have affinity for the same antigen, those antibodies are called as the monoclonal antibodies. If the antibodies are specific to one epitope, they are still called as the monoclonal antibodies. If they are specific to one type of cell or tissue, they are still called as the monoclonal antibodies. So this term monospecific can refer to the specificity to the same antigen, to one epitope, or to one type of cell or tissue. When you talk about the antibodies, this term monospecific and the monovalent, they actually overlap in meaning, and they refer to one and the same thing. And this means that the antibodies, they are made by identical immune cells, and they are clones of a unique parent cell, in contrast to the polyclonal antibodies, which are made from several different immune cells. What is the difference between the monoclonal antibodies and the polyclonal antibodies then? If you look at this particular diagram, this is an antigen, and on the surface of the antigen you can see these different kind of the epitopes. This one is in the yellow color, this one is in the blue color, the purple one, the orange one. So you are actually having a different kind of the epitopes on the surface of this particular antigen. Now the monoclonal antibodies, what they do is that they react with a specific epitope on a given antigen. Like in this particular diagram, if you see, you have got 1, 2, 3, 4, 5. You have got 5 different types of the epitopes, but these particular antibodies, they are only interacting with one of the epitope. Therefore you call them is the monoclonal antibodies, because they are interacting with one epitope out of the 5 epitopes that are present on the surface of this particular antigen. Now, these monoclonal antibodies, they are made from a single parental cell. They are clones of a single cell. When you talk about the polyclonal antibodies, as you can see over here, some antibodies, they are interacting with this purple epitope. Some of the antibodies, they are interacting with this blue one. Some are interacting with this yellow, the orange one. So different antibodies, they are interacting with different epitopes present on the surface of this particular antigen. Therefore we call them is the polyclonal. And this polyclonal means that these antibodies, they are not made from a single B cell. They are made from different B cells, and they react with various epitopes that are present on the surface of this antigen. Now, given almost any substance, it is very much possible to produce monoclonal antibodies that specifically bind to that particular substance, and hence that particular monoclonal antibodies can be used to detect or purify that particular substance. Like for example, if you have got this antigen, and if you put that in a mixture, and if you use this particular antibody, this antibody is going to attach to this particular epitope. Therefore it can detect or purify this particular antigen among the mixture of the antigen. And these monoclonal antibodies, they have become an important tool in biochemistry, in the molecular biology and medicine for a variety of the purposes. For example, in the medication, in the diagnosis, for example. When used as medications, the non-registered or the non-proprietary name for these monoclonal antibodies usually ends in the MAB. So this is actually you can say a drug, a non-proprietary or the non-registered name used at the end of the monoclonal antibodies. If you talk about the discovery of these monoclonal antibodies, so the idea started at the beginning of the 20th century when Paul Ehrlich he gave the concept of the magic bullet, and he postulated that if a compound could be made that selectively target a disease-causing organism, then a toxin for that particular organism could be delivered along with the agent of the selectivity. For example, if you have got an antigen, and if you can design a monoclonal antibody that specifically interacts with that particular antigen, and if you attach something to this monoclonal antibody, this monoclonal antibody can actually work as a delivery man to deliver the drug to that particular antigen, therefore killing it. One of the specific work he and the Eli Machnikov, what they did is that they received the 1908 Nobel Prize for Physiology and Medicine for the effective treatment of syphilis using this particular strategy, that they designed a particular compound, and that compound was specifically interacting with the bacteria that was causing the syphilis, and to those particular compound they were actually attaching a particular drug, and that drug was killing that particular bacteria, therefore treating the syphilis. And this treatment that was actually there in the 1910, and for this particular work they were given the Nobel Prize in 1908. Now in 1970, the B-cell cancer, commonly known as the multiple myeloma, that was known, and it was understood that these cancerous B-cell, all of these, they produce a single type of the antibody, and when they are producing a single type of the antibody, they were actually using them to study the structure of the antibodies. But up to 1970, it was not yet possible to produce identical antibodies against a specific antigen. So the technique of the hybridoma, that was still not there. In 1975, Kohler and Milstein, they succeeded in making fusion of the myeloma cell lines with the B cell to produce hybridomas that made antibodies to known antigens and that were immortalized. You will get the concept of this immortality and the production of the antibodies in the coming slides. And they shared the Nobel Prize for Physiology and Medicine in 1984 for the discovery of these hybridomas. Now, how these hybridomas, they are actually produced, how you are actually going to produce a hybridoma against a specific antigen. Now, the monoclonal antibodies, they are typically made by cell culture technique that involve fusing myeloma cells with the spleen cells from mouse that has been immunized with the desired antigen. So there are three things that you need to understand. The first thing is that you have to inject the desired antigen into the mouse, and when you inject the desired antigen into the mouse is going to produce antibodies only against the antigen which you have injected. That means your desired antigen. This is one thing. Another important thing is that in the production of the hybridoma, the myeloma cells that you are using, they are deficient for an enzyme which is known as the hypoxanthine guanine phosphoriboside transferase. This is an important thing to keep in mind, that the myeloma cell, they are actually deficient for this particular enzyme, and we will come to the importance of this enzyme in a while, that this is a very important enzyme when you talk about the salvage pathway of the DNA synthesis. The third thing, that the spleen cells that you are using, they are actually having a normal copy of this particular enzyme. So they are not deficient for this particular HGPRT enzyme. This means that the spleen cell have the ability to carry on the salvage pathway. So these are the three important things to keep in mind, that the mice that will be producing the antibodies against your desired antigen, the myeloma cell that will be deficient for the HGPRT, and the spleen cell, they will be carrying a functional copy of the HGPRT enzyme. Now recently what you can do now is that you can also use the rabbit B cells to form a rabbit hybridoma. So initially they were using the mouse cells to make a hybridoma. Now you can also use the rabbit cells for the formation of the hybridomas. For the fusion of the myeloma cells to the spleen cells, you are actually using the polyethylene glycol, and this polyethylene glycol is actually fusing the adjacent plasma membranes, but the success rate is very low, so you have to use a selective media so that you can actually focus on these hybridoma cells and not on the other types of the cells. This is a very general diagram how you are actually going to make these hybridomas, that you will be injecting the mouse with your desired antigens. The mouse, they will be producing the antibodies utilizing their spleen cells. You are going to mix the myeloma cells which are deficient for the HGPRT enzyme. You are going to combine them with the spleen cells, and when you combine them, you are actually going to have these hybridomas. You are actually fusing the myeloma cells and the spleen cells to get these hybridomas. As you are actually fusing two cells, therefore you are calling them as the hybridomas. When you fuse the myeloma cell to the spleen cell, you are going to get a variety of the cell that we will discuss in the next slide. So what you have to do is that you have to use a selective media, which is called is the Haight media, to harvest these monoclonal antibodies. So this is a general overview of the process of the production of the monoclonal antibodies. When you fuse the spleen cells and the myeloma cells by using the polyethylene glycol, you can actually expect 5 kind of the cells. The first one is the fused B cells, and this one is fused B cells. That will be one of the type of the cell that will be there when you mix the B cells. The plasma and the B cells, they are referring to one and the same thing. The B cells and the plasma cell, they are one and the same thing. So one of the cells that you can get after the fusion is the fused plasma cells, that the polyethylene glycol that is actually fusing the two plasma or the two B cells, giving you the fused plasma cells. You can also have the fused myeloma cells. You can have the unfused plasma or the B cells. You can actually have the unfused myeloma cells, and then you are actually having these hybridomas. So you are not interested in the fused plasma, the fused myeloma, the unfused plasma or the unfused myeloma cells. You are only interested in the hybridoma cell, because these are the cells that is going to give you the desired results. Now, how to select this hybridoma then out of these 5 types of the cells? To select this particular hybridoma, you have to use a selective media, and this selective media is called is the Haight media, which is actually the hypoxanthine aminotyrine thymidine media. Before going into the media, you need to understand that there are 2 pathways for the synthesis of the DNA in the living organisms. One of the pathway that is called is the de novo synthesis. Now the de novo synthesis, that is actually the synthesis of complex molecules from the simple molecules. Say for example, if you are using the sugar to make the polysaccharide, the sugar to make the DNA, for example. So if you are using the synthesis, you are making the complex molecules from the simple molecules. That particular phenomena is known as the de novo synthesis. So one of the phenomena for the synthesis of the DNA, by that I mean for the synthesis of the nucleotide, that is known as the de novo synthesis, in which you are making the nucleotides from the simple molecules. One of the important enzyme for this de novo synthesis, that is known as the dihydrofolate reductase. just keep in mind that this particular enzyme is one of the important enzyme for the de novo synthesis, because this is one of the key enzyme which the Haight media is using to select the hybridomas. Another important pathway is the salvage pathway. And the salvage pathway, a biological product is produced from the intermediate in the degradative pathway of its own or a similar substance. That means you are not using the simple molecules, you are actually using the intermediate of the degradative pathway for the synthesis of the complex molecules. And the most important enzyme for the salvage pathway is the hypoxanthine guanine phosphoriboside transferase. This is one of the important enzyme. Now forget about this minus over here, but the important enzyme for the salvage pathway is the HGPRT, and the important enzyme for the de novo synthesis, that is the dihydrofolate reductase. Now that doesn't mean that these are the only enzymes that are required for the de novo synthesis or the salvage pathway, but I am focusing on these two enzymes because these are the enzymes which the Haight media used for the selection of the hybridomas out of the other cells. Now this Haight media, this H is for the hypoxanthine, this A is for the aminopterine, and this T is for the thymidine. Now this Haight media, it is a selection medium for the mammalian cell cultures which relies on the aminopterine. This is one of the component of the Haight media, and the function of this aminopterine is to block the activity of the dihydrofolate reductase. And if you see this, I've told you that this dihydrofolate reductase is one of the important enzyme for the de novo synthesis. And if you are blocking this enzyme, the dihydrofolate reductase, that means you are blocking the de novo synthesis of the DNA. The other two component of the Haight media, one is known as the hypoxanthine and the other one is the thymidine. These two are very important when you talk about the salvage pathway of the synthesis of the DNA. Now, how this particular media works, how this is going to select the hybridomas out of the other types of the cell, let us discuss this. When you talk about the unfused and the fused myeloma cells, you can have the unfused myeloma cell, that was one type of the cells, and the fused myeloma cell, that was the other type of the cell. Now they cannot grow on the Haight media because they lack the HGPRT and thus cannot replicate their DNA. If you talk about these cells, like about these fused myeloma or these unfused myeloma cells, they can go for two types of the DNA synthesis. One is the de novo synthesis and the other one is the salvage pathway. Now in both of these cells, in all of these cells, you have blocked the de novo synthesis because you are adding this aminopterine. That means that all of these cells, they will not be able to go for the de novo synthesis because the aminopterine is blocking the activity of the dihydrofolate reductase. So when you talk about these fused myeloma cells or these unfused myeloma cell, they are not going to grow on the Haight media because you have blocked the de novo synthesis by the aminopterine, and as they are deficient for the HGPRT enzyme, they are deficient for this particular enzyme. So, and this particular enzyme is very important for the salvage pathway. So the fused myeloma and the unfused myeloma cell, they cannot have the de novo synthesis, they cannot go for the salvage pathway, therefore they cannot grow on the Haight media, therefore they will die out on this particular Haight media. When you talk about the unfused and fused spleen cells, they cannot grow indefinitely because of their limited lifespan. In the unfused and the fused spleen cells, of course, there will be no de novo synthesis because the aminopterine have blocked the de novo synthesis, but these spleen cells can utilize their functional copy of the HGPRT to go for the salvage pathway. But as their lifespan that is limited, therefore they cannot grow indefinitely on the Haight media, and hence they also die out on the Haight media. So you have removed the fused plasma, the fused myeloma, the unfused plasma, and the unfused myeloma cell on the Haight media. The only cell type that can grow on the H media, they are these hybridoma cell. Now how these hybridoma cell, they can actually grow on the H media. Now if you talk about this hybridoma, this is actually the combination of the B cell and the myeloma cell. So in the hybridoma, you have got the B cell as well as the myeloma cells. Now the de novo synthesis that have been blocked in the hybridoma because of the aminoterine, but as in this hybridoma there is a B cell, and the B cell have a functional copy of the HGPRT. So the HGPRT that is going to go for the salvage pathway, and the myeloma partner that is going to provide immortality to this hybrid cell. Like if you talk about this hybridoma, you have got the spleen cells. So the spleen cell is going to supply a functional copy of the HGPRT. So now this hybridoma, it can go for the HGPRT utilizing the hypoxanthine and thymidine as the intermediate, and the myeloma cell as they have got immortality. So the myeloma cell that is going to make this particular hybrid cell is the this particular hybridoma like the cancer cell, and it will grow indefinitely on the H media. And we will continue this discussion in the next video.